So good morning, everyone. Thank you so much, really, for having me here. It's a pleasure for me to get out of my little cocoon and dark corner over at Cyber in the lab and working all the time. So I'm really, really honored to be here. Thanks, Rich, for the invitation. My talk today is about malformations of cortical development and some of the work that we've been doing on this end. I'm a geneticist by training, so a lot of it is going to be based on genetics, but I think what's really exciting, certainly for us geneticists who hardly ever treat patients, is that there's actually been a lot of progress in the area that I'm going to talk about, where there are really good therapeutic options for kids on the horizon here. And I know that some of our very close collaborators in the room, Jason, Jeff, have been really on the front line of this as well. So really briefly first, the problem we study over here, which are malformations of cortical development. These are really early developmental problems that occur in the developing human brain. Typically, these originate in the fetal period, very early fetal period, and they encompass a spectrum of disorders. And some of these images, which I acknowledge are a little bit on the small side, some of these are really like classic brain malformations, like classic lissencephaly, for example. And then you have a bit of a variable spectrum beyond that, which includes polymicrogyria, megalencephaly, and focal cortical dysplasia spectrum. Mid-hindbrain malformations that include cerebellar malformations like Dandy Walker and others. And so, traditionally speaking, these were all classified, and in clinical practice also, they were really classified and categorized and grouped based on the stages of neuronal development during which we assume the abnormality occurred. But I think what's been really exciting, and while this is extremely useful, is that the genetic technologies that came around about 10 years ago, and where we started to do a lot of testing for kids, have really shown us that a lot of these malformations are caused by perturbations in major cellular pathways. And when you talk about cellular pathways, then you're a step closer to thinking about therapeutics rather than thinking about just very isolated, fundamental, biological processes. So we now know that there are pathways and networks that are disrupted across groups of these conditions. And again, that brings us closer to therapeutics and a lot of discussions with pharma in the process. So one question could be is why study malformations of cortical development? I get this question from my colleagues in genetics all the time. These are ultra rare disorders, aren't they? So why, why do we want to really figure out the genetic causes for these things? And another really, I think sometimes false assumption is that these disorders are so devastating neurodevelopmentally that there's very little we can do. And I think that is also sometimes an overgeneralization. So malformations of cortical development collectively are commonly seen in children with intellectual disability, autism, epilepsy, feeding issues, and tone abnormalities. And what's really interesting is if we look at these as isolated, disorders or conditions, you really miss, I think, we miss the big picture in a way, which is the fact that there's a really broad spectrum, and kids vary quite a bit. So some kids, for example, with for example, brain overgrowth, some kids are really milder than others, and they are in the school system, but at the same time, they still have important developmental problems that often go undetected, unassessed, and things like that. Importantly, and this is, I hope would be of interest, is that one of the main conditions we've been studying has been focal cortical dysplasia. And as this audience may know, this is among the most common causes of focal intractable epilepsy in kids. Some studies assess up to 50% of children with undergoing epilepsy surgery. Malformations of cortical development as a group are seen in more than 500 genetic syndromes, again with very little, sorry if I'm in your way by the way, very little understanding about how to manage these children. The diagnosis, the medical management across the board are not uniform for these kids. So oftentimes we deal with families who are extremely frustrated by their children's care. Now in terms of the etiologies for these, to be fair and not to be completely focused on genetics, there are a lot of non-genetic etiologies as well. Certainly, disruptive acquired injuries during pregnancy, vascular insults that a mom may be exposed to are really important causes. Stroke, fetal stroke, for example. Infections like Church's infections. Zika virus was, of course, a really famous recent cause. Teratogens and toxins, and then other causes of maternal deprivation. Even untreated maternal metabolic disease can cause malformations of cortical development. I'm going to really focus on, of course, the genetic causes of this, but with a really special focus on a specific pathway. And so one thing that we have appreciated a long time ago is that malformations of cortical development can be quite unique. So anytime we're talking about the developing brain, it can actually be susceptible to its own unique set of genetic mechanisms. Generally speaking, across the board, when we talk about genetic mutations, the most common scenario is that a genetic mutation originates very early on, either in an egg or a sperm at the time of fertilization or shortly before fertilization. And therefore, and this is shown in the green color over here, and so basically what you see is that the mutation is present in almost in all cells of the fetus. So this is called a germline mutation or a constitutional mutation, and what that means is that then the kid will have the mutation everywhere. Usually these kids have a wide variety of genetic syndromes. For MCDs in particular, we've noticed that there's another mechanism that comes into play here, which is that sometimes a genetic mutation can originate later on during development. It can be at the 2-cell stage, it can be at the 4-cell stage, and as a result, a child can actually be mosaic, meaning that some cells will have the mutation and some cells will not. And this is actually an emerging spectrum now, because our technologies have gotten better at detecting mosaic mutations. We're now seeing them more. And so the same thing applies to the developing human brain. And so what's been interesting, and this is a really growing, I think, body of literature that's really quite fascinating to follow. So lots of groups, including Chris Welch's group in Boston and others, have shown that a lot of malformations of cortical development can be mosaic, depending on which stage. So this is actually shown, by the way, here. So if it's at an early stage where the mutation originated, then usually you'll get it again in all cells. Whereas if it actually occurs in the spermatocyte here, which is shown here, you'll get it as well in all the cells. And this is a good example of lissencephaly. In double cortex syndrome caused by DCX, for example, we have seen mosaic mutations where you can see the subcortical band here. It can originate later on in development. And then, of course, hemimegalencephaly is the poster child. People think of this as the poster child of mosaic mutations because you clearly see it on the MRI scan that it's extremely focal and it originates naturally later on during development. So we know that when it comes to these brain malformations, that mosaicism can occur. It can really occur actually very broadly at any stage during a human development, in any dividing tissue. In any species, actually. It's been reported across species, and it can occur either at an individual level, in vivo or in vitro, in the lab, in culture, when we're working with these with cell lines from kids and such. So it's a really important phenomenon. And this is another paper, actually, that was also published by Chris Welch and Ann Feduri at the time, that actually showed that mosaicism is actually seen in a lot of malformations of cortical development. And these are just some of those examples. Again, hemimegalencephaly and FCD are prominently represented in this topic, and then heterotopia, periventricular nodular heterotopia, and others. So really the brain we think of now as a mosaic structure, essentially. So now the question is why is a molecular diagnosis important to begin with? And this is, again, a question we deal with pretty much all the time. I think it's really helpful for natural history studies, disease-specific medical management for these kids, again, because currently it's all across the board because of the rarity of the individual conditions. Molecularly targeted therapies, which I'll talk about, and then, of course, needless to say, recurrence risk. And a lot of insurance companies don't even acknowledge that this is important, that this knowledge is important for the family, and that's another topic altogether. So the group of disorders that I would like to talk about today are things that this audience, again, will be super familiar with. But this is a spectrum of brain overgrowth phenotypes that we call the megalencephaly spectrum, just for simplicity's sake and to package it in our heads a little bit. This is a spectrum of brain involvement that includes diffuse brain overgrowth without necessarily cortical dysplasia, and a lot of these kids have neurocognitive problems. Then you can actually have diffuse cortical dysplasia, what we've called dysplastic megalencephaly. And then you have the more focal spectrum over here that ranges anywhere from hemispheric involvement in classic hemimeg, quadrantic involvement in what was called previously as quadrantic dysplasia, and then focal cortical dysplasia, which is, of course, last but not least, because it's an extremely common cause of epilepsy requiring epilepsy surgery. So what we've now known through work over the past 10 years is that this entire spectrum that I just showed you are caused by mutations of this cellular network. And for those of you who have been in this field, this is the mTOR pathway. This is my cartoon figure of it. There's like thousands of them in the literature. And the mTOR pathway is a really fascinating pathway because the oncology groups in the world have really been all up in this pathway because of the fact that it's commonly mutated in somatic tissues in cancer in adults, in a wide range of cancers, in adults and kids, actually. And so to summarize really a lot of literature that's quite relevant to FCD and hemimeg is that we know now that there is a spectrum of pediatric phenotypes that include brain and body that are caused by mutations of this network. Usually the mutations, and I think this is a really key point, all these mutations have the same functional endpoint, which is that this pathway, which is a growth pathway, is hyperactivated. So most of the mutations result in gain of function of the pathway function, essentially. And that's really key. The mutational spectrum overlaps significantly with that's seen in somatic tissues in cancers. So whether it's pediatric population with developmental problems or adults with cancers, usually the mutations, if you compare across these two groups, the mutational spectrum is the same. And that's really a really important point, because it places us for the first time in a really great position to think about therapeutics, because this pathway has been targeted by pharmaceutical companies for decades now for cancer indications primarily. So the famous example, of course, is sirolimus or everolimus or rapamycin. So for the first time in a long time, we have these severe fundamental brain disorders that are actually potentially treatable using some of these inhibitors that have been developed for other indications. So I think this is really interesting. And so when we look at this pathway, the mutations actually range. So it's not just one gene. This is actually all the red stars are showing you all the genetic components in which we have identified mutations in children with brain phenotypes. And so this is these genes. Some of them are upstream in the pathway. Some of them are midstream, such as the AKT complex right here, which is like the hub of the pathway. And then you have a few downstream ones, including the infamous mTOR itself within the pathway. And that's really interesting. So there's a lot of heterogeneity. And I think geneticists, and I'm guilty of this as well, sometimes we box ourselves in where we think about one gene at a time or one mutation at a time. But if we do that for our children, we do run the risk of missing the bigger picture and trying to really impact, again, their therapies and medical management in a more global perspective. So we kind of have to shift our way of thinking a little bit from a genetics way to more of an oncology way, where we think about networks as a whole. And mutations can be either activating mutations of activators of the pathway or loss of function mutations of inhibitors of the pathway. So you can have two mechanisms, but they all result in the same thing. You're basically cranking up this important cellular network. And so it's working over time, usually in these overgrowth phenotypes. What I'd like to show from this, to be honest with you, is that what's really fascinating is that when people talk about successful diseases that have gone all the way from gene discovery to therapies, usually they show a wheel that has sometimes these components. When you talk about natural history studies, preclinical models, diagnostic approaches and biomarker discovery and clinical trials. And what has been really a true pleasure for me here in Seattle is that collectively as a group, here between SCRI, UW, the Hutch, between our teams, including neurosurgery, neurology, and genetics, we pretty much have ongoing work in all of these areas. So it's been truly exciting to be here and to be involved and help kind of address one peg of the wheel at a time sometimes. So with that, I'm going to actually show you just some examples of these phenotypes and then show you some novel findings from our research in the hopes that it's useful. So first, I'll show you some examples of mutations of this gene, PIK3CA. This is probably the most common. Mutations of PIK3CA so far have been found in the entirety of that spectrum that I just showed you, where there is anywhere from diffuse brain overgrowth all the way to focal cortical dysplasia. Maybe I should stand over here. And so equally important is that kids can have an equally diverse spectrum of body overgrowth phenotypes. So including, actually, we believe sometimes intracranial AVMs. But the body phenotype can range quite a bit from really dramatic syndromes, such as this is known as Clove syndrome, to what's been known as Klippel-Trenaunay syndrome, to focal, what's called macrodactyly, where there's isolated overgrowth of digits, and then facial lipomatosis and lymphatic malformations. All of these phenotypes have been caused by mutations of PIK3CA. So, and if you look at the literature, each of these entities, there's 10 clinical entities, describing these phenotypes. Oh, thank you. You're gonna have between two ferns kind of. So essentially, if you look at the literature, these are all the diagnostic entities that again have been described as distinct disorders, and these are for the body. And so it's really the case of the blind man and the elephant, where depending on which group is publishing on what are they studying, they're reporting these as distinct disorders. They're all caused by mutations of the same gene, which is PIK3CA. So again, when we talk about therapeutics, we can really be much more empowered, I think, if we think about this as a more common problem than just thinking about ultra-rare entities. And so importantly, what we like to think of it is to say, where is the mutation? At least for me, this has been a more useful frame of mind when I see children with PIK3CA mutations. I think it's really helpful to see where is the mutation present, which tissue of this child is affected. And I think based on that, we can predict certain outcomes. So importantly for the brain, It depends also on whether they have cancer, activating cancer mutations, or non-activating cancer mutations. The kids with activating cancer mutations tend to have the hemi-negative FCD, more severe brain phenotypes, severe intractable epilepsy. Usually they're at a lower risk, generally speaking, for hydrocephalus and Chiari malformations, which are important neurosurgical complications in the kids who have PIK3CA mutations that are not activating. And I'll show you some examples of that. So kids in this group tend to have a higher risk for hydrocephalus, higher risk for Chiari. Usually they don't have a lot of cortical malformations, actually. They have a diffusely overgrown brain, but not a lot of cortical dysplasia, generally speaking. And so if they have epilepsy, it's usually really mild. And we'll leave the body phenotypes for another day. So PIK3CA mutation, or PIK3CA related overgrowth, which is abbreviated as PROS, has been known to be associated with hydrocephalus across the literature. We don't really know what the actual risk is, but in one of our earlier studies where we looked at smaller cohorts arguably of these kids, we noticed that ventriculomegaly was present in 81%. Again, these ends are small, as you can appreciate, with frank hydrocephalus requiring shunting in about 40%. So this is really, I think, it's more common than certainly a lot of genetic syndromes that we deal with. And so that's really important. The other neurosurgical complication in kids with milder mutations has been Chiari malformations. And these are just some representative images here showing how it can look like. And certainly we've seen this in a growing number of kids as well. And we don't also know the actual risk, but we've seen some degree of ectopia, cerebellar tonsillar ectopia, in 27 out of 40 at the time. A number of kids can require posterior fossa decompression. So really important, I think, neurosurgical considerations. So moving on from PIK3CA, I just wanted to briefly show you its kind of sibling here is PIK3R2. Mutations of PIK3R2 are associated with a syndrome known as MPPH syndrome, which stands for megalencephaly, where there's diffuse brain overgrowth, polydactyly, which is shown over here, sorry. There's the extra digit. PMG, which you can appreciate is quite diffuse, and hydrocephalus. So this is also caused, this is an important hydrocephalus syndrome in the genetics world at least, that's caused by mutations of this pathway. Moving on, this is AKT, or AKT3 specifically, and this is at the hub of the pathway. So this is really interesting because AKT3 is considered the master regulator of this whole network. So when we talk about therapeutics, there are inhibitors of AKT specifically that I think we really need to be careful about because you don't want to knock out the whole pathway, and AKT3 is the master regulator of the pathway. And so mutations of AKT3 are associated with a very similar spectrum that I showed you with PIK3CA, with PIK3R2, where there's diffuse polymicrogyria. This is a girl who some of you may know. This is one of our local Seattle children's patients who had an AKT3 mutation, and she had this vascular lesion over here that was quite striking. And so we see these vascular things sometimes. They kind of help clue us into the pathway, but not always, But this is what she had. This is a child who had an AKT3 mutation who had diffuse brain overgrowth, severe epilepsy, died suddenly during sleep, assumed to be due to SUDEP, and the family donated his brain to research at the time. So it was a 5 a.m. call where we needed to coordinate getting this full brain from Florida, and when we studied this brain, it was quite fascinating, and Bob Hefner at the time was really, really helpful with some of these studies, but the brain weighed, essentially, at 8 years of age, weighed double the size of an adult. So it was quite large, and there was diffuse polymicrogyria on neuropathology, and it was large enough that we actually wanted to see what is the largest brain size documented in humans. And so at the time, we went on this interesting intellectual journey of looking up the largest brain size documented at the Smithsonian. And this is the graph that's actually showing you the green columns or bars are actually the largest brain sizes that have been documented by weight over here. And these are all adults. The red bar is our child. So he actually was larger than any of the documented brain sizes for adults at the time. So we know that this, and we've known this, by the way, from P10 before. P10 is the other gene within this pathway that has been published, for decades. We know that mutations of this pathway can cause really extreme forms of overgrowth, but this was kind of a reminder how severe it can be and how big these brains can be. And it's really remarkable he survived for as long as he did. For the girl that I showed you, so this is the girl with the AKT3 mutation. We had assayed multiple brain tissues from her surgeries at the time, and this is the AKT3 mutation that she had. This is the level of the mutation, which means how mosaic it was in the brain. So you'll see it ranged from 10% to 18% in the affected brain regions that were removed surgically. What was interesting, and this is more, to be honest with you, useful sometimes for geneticists and pediatricians trying to diagnose these kids as early as possible, is that when we assayed her skin, so we couldn't find this mutation in blood or saliva, by the way. So we tried to assay the skin to see if we can detect this mutation. It was at an extremely low level, 1.3%, which means that there's a really high value in doing molecular studies on brain tissues when possible. Because oftentimes, if we try to spend a lot of time and energy testing skin or blood or saliva, we're just not going to miss it. This is never going to be detected, certainly not using any of the standard clinical lab panels. This is all through research testing where we have the luxury of trying to dig deeper as much as possible, but clinical testing will not detect this. So it was really interesting. We could only find the mutation, technically speaking, when we assayed the brain tissue, which was quite helpful. And then last but not least is the infamous mTOR itself. So mutations of mTOR, this is actually quite interesting because now we know that this gene is the most common cause so far of focal cortical dysplasia, the most common genetic cause of focal cortical dysplasia in children. Usually mutations of mTOR in these kids are very low level mosaic, meaning that the level in the brain can be, depending on the extent of involvement, can be anywhere from 0 to 10%. It is virtually never detectable in peripheral samples. So if we see these kids in clinic again and we try to send off clinical testing, we will not be able to pick up these mutations, generally speaking. But there's a brewing spectrum of mTOR-related disorders. So it can actually be sometimes higher level mosaic and can cause bilateral malformations, like bilateral PMG, for example, and these pigmentary skin lesions that can be a useful hallmark. For some of you who may have tracked the hemimegalencephaly literature, these resemble what's called a hypermelanosis of E2, which are these very striking hyperpigmented linear markings that kids have. And this has been reported as an association with hemimegalencephaly before in the literature, and we think it's all caused by mTOR mutations. And then interestingly, actually, we can have germline mutations of mTOR causing diffuse brain overgrowth with autism, just like P10. And these kids usually are not neurosurgical candidates. This is a child that we actually studied together with Jeff, and at the time, this was more than 3 years ago, who actually had focal cortical dysplasia and underwent surgical resection. And we found a mutation in mTOR, which is actually shown over here in this region. So this is where we exactly got the sample where we got the DNA from. And we noticed that it was present at 8%. When we did immunostaining for phospho-S6, which is a downstream marker of the mTOR pathway, and we noticed that it was elevated, suggesting gain of function or activation of the pathway. And interestingly, you can appreciate here, of course, there are EEG abnormalities that were noted at the time. Not unlike a lot of the other children, this patient had recurrent seizures after surgery and went back in for a more extensive resection. So what we did at the time is then we assayed again. We sampled any additional pieces of tissue that are available, and we looked for the same mTOR mutation. And what you'll notice is that it was at a lower level, between 0 to 2% in the more perilesional tissue. Phospho-S6 was elevated, but it was less elevated than with the first lesion. And the degree of EEG abnormalities, they were there, but they were less severe than certainly the first focus that was surgically removed. So what this kind of suggested to us for the first time was that perhaps there is a link between the genetic mutation, the degree of pathway abnormalities, and the EEG abnormalities and morphological abnormalities on the MRI scan. And this was the first time we kind of saw that correlation, because most studies, again, rely on assaying one tissue, they find a mutation, and then they're done. And so this was quite fascinating because this adds the translational value of all that work. So, and I certainly don't think that our goals should just be focused on just finding the mutation and be done with it. I think we should really try to correlate that with what the child has. So this is just a really interesting patient that I think we learned a lot from. So to summarize really like what we've learned so far, and this is based on, in the 7 years that I've been in Seattle, we've basically studied about 350 children. We basically, on the research side, we will take any sample we can get. So we are extremely liberal because we know that testing clinically is fraught with all sorts of challenges. And what we've learned is that there's a spectrum of brain phenotypes, and depending on which spectrum, you'll have different levels of mutation. So with the diffuse ones, the kids with the diffuse brain overgrowth, usually they'll have megalencephaly. You should be able to pick up the mutation in peripheral tissues and in other affected tissues. So these are usually germline mutations seen at 50% level. With hemimegalencephaly, usually we see these mutations mostly in the brain, and these are the levels of mosaicism across the board, across the three most common genes. But you can detect it in potentially peripheral tissues, potentially. And then finally, and certainly last but not least, with focal cortical dysplasia, these mutations are virtually almost always only detectable and really difficult to detect in peripheral tissues. So the key questions we have for the second half of my talk is, well, where do we go from here? Can we develop better diagnostic tools now for our kids? Because not all, having research testing options is not also ideal for these kids, because to be eligible in clinical trials, a lot of the time you need them to have a CLIA CAP certified result, you need them to be genotyped appropriately and stuff like that. So how do we better improve the diagnostic end? And then, importantly, should we be thinking about using mTOR inhibitors for these kids? And so these are the two questions our group has been involved with to some extent. So on the diagnostic side, I have to say, I think one of the biggest issues is tissue availability, naturally speaking. So ideally, we want to test affected tissue. And this is one of those things where here in Seattle, I think it's been one of the biggest privileges actually working with our neurosurgical team here, because of the fact that we have the possibility of doing that. And a lot of centers across the country have not been able to do these things. There's a lot of logistical issues to overcome, as you may appreciate. But this is the ideal situation, is that you need to test lesional tissue, ideally in these children. And then the sequencing sensitivity must be there. So you need to use the testing method that gives you the adequate sensitivity to detect these. And so some of the common clinical platforms, like standard Sanger sequencing or exome sequencing, which is considered the top-notch genetic test, may actually lack the depth of coverage that you need. So if you get 30x, which means that you get 30 reads across your region of interest, if you have a mosaic mutation in a child at 1%, it will not be detected at this level. So this is, I think, really important kind of considerations when we're testing these kids and thinking about designing clinical trials. So these are the two, I think, balancing acts we kind of have to keep in mind. So with that, I'm going to share some unpublished data, just to kind of show you where, new findings, in the hopes that they're interesting. So our group over at SCRI, our genomics team, follows a tried and true approach, basically, for these conditions. We start off with targeted testing for mTOR pathway genes and mutations, the known genes. If they're negative, then we will reflex to exome sequencing and now genome sequencing. And then we do, really, this is where I'm mostly interested, to be honest with you. It's not, the fun part, the challenging part is not finding the mutations, is where do we go from there? So genotype-phenotype correlation studies, functional assays to understand what these mutations do. These are the areas that I hope will never go out of style, essentially. What I wanted to highlight briefly is that we use a paradigm that is a little different than most genetic groups that don't study these conditions, which is that typically most study designs rely on analyzing mom, dad, and then the child, and usually it's one sample from the child, right, where they aim to find the genetic cause. But we know now that for FCD, Hemimeg, and related disorders, we actually need to look at affected tissue. So we use what's called a paired approach for each child. We get a brain piece, if possible, we try to test the brain tissue, as well as a peripheral sample, whether it's blood, saliva, whatever we can get our hands on, basically. And so this is a paired approach that our group has been using. So with that, about 3 years ago now, we decided to use this method. And for those of you who work with, with brain tumors and things like that, this is digital Drop of Pcr, which is now a popular method in cancer diagnostics. Digital Droplet PCR is essentially a very sophisticated form of PCR where your PCR reaction is fractionated. Your reaction is fractionated into 20,000, ideally, droplets. If you've got good hands in the lab, you'll get 20,000 droplets generated per reaction. So essentially, you've got 20,000 readouts from the same DNA molecule. And so what that allows you to do is actually really do ultra-deep sequencing. So you get 20,000 readouts for every single molecule that you have in there. And so what we did is that we designed a very basic panel. We call it the mTOR hotspot panel. We took the most common mutations associated with focal cortical dysplasia. These are also the most common mutations seen in cancer. So we call this the hotspot panel. 6 mutations, which are by far more than 80% of all mutations seen in these kids. And we tested pretty much all the kids we had and all the samples we have in the research lab. This included a total of 151 kiddos with a total of 332 brain samples. And this is a really valuable brain tissue repository that was started by Laura Jensen at the time here and others. And so this is a really good repository, good sized repository. So we found mutations in 14% of kids. And for those of you who may say, well, this is a really low yield, and I get that, I think it is on the lower side, but what I would remind folks is that this is just looking at 6 mutations. So it's just very specific, it's a very specific targeted panel for these kids. And for a yield, so it's a very simple test, and for a yield of 14%, I do think it's worth considering such an approach as an initial first-tier test. And so this is a breakdown of the mutations that we found in the different genes. And they showed us a number of really interesting insights, and I'll show you these as case examples real quickly, 3 examples specifically from patients. So looking across the board, and I just wanted to show this kind of massive plot here, across all the tissue types we got, and this is showing the level of the mutation as a percentage. What you'll notice is that most of them cluster in their lower range. So this is the 20% line over here. So most of the mutations cluster below this, which means that these mutations that are falling within this zone over here will be missed by standard clinical sequencing. It's only a minority of the kids who have really high-level mutations that will be detected by standard clinical tests. So this is just one of the main takeaways for us from this experiment. So now I'll show you briefly 3 quick examples of case presentations of kids. So this is a child who was evaluated at Seattle Children's Hospital full term, had a large head size of +4 standard deviations, and very early onset of tractable epilepsy, of course, and underwent hysterectomy at the time. And when we did digital drop of PCR, we found one of the common PIK3CA mutations. Interestingly, the mutation was present at 20% in affected brain tissue, but, and this is where one of the reasons we wanted to show this, is that the mutation was detectable in saliva, which is one of the first cases where we saw that we can actually detect it. I think this is an exception rather than the rule, but still, this is quite interesting. And we don't understand it just yet developmentally. How could a child have a mutation present at 20% in these 2 very different tissues? So this is a digital droplet PCR plot, which is a 2D plot that shows you where the mutant droplets, which are shown in blue here, cluster. So these are the mutant droplets, these are the wild type droplets. And based on the software program, it gives you basically an estimation of the level of mosaicism, so it was 20%. So that's this child, and this is just showing you here more the number of droplets we got from these samples. The second case that I wanted to show you actually, this is a case that's similar to the mTOR case that I showed you earlier. So this is a child who had diffuse, as you can appreciate, diffuse dysplasia and underwent extensive resection, and histopathology showed FCD type 2A, which is among the most common types that we've seen in our cohort. So we found this mTOR mutation, which is among the most common mTOR mutations. That's the same mTOR mutation in the previous child that I showed you who had surgery. And what's interesting is that when we look across, we had about 10 brain tissue samples from this kid because of the extent of the resection. And so when we look at the level of mosaicism, it's so variable, and it ranges anywhere from 0% in some brain tissue to up to 14%. And this is a tabulated version of this, so it shows you the tissue and the region of the brain specifically, the number of mutant droplets, wild-type droplets, the level of mosaicism shown over here. And so this was really fascinating because not a lot of groups have been assaying molecularly a number of tissues from the same child. This is, again, one of the advantages that we've had working in Seattle, is that we have had, because of this amazing collaboration, we have been able to get multiple brain regions from these kids and really have a broad overview. So the question we asked, well, does this level of mosaicism, does the level of the mutation correlate with neuropathology? And so we had, at the time, Dr. Raj Kapoor from Seattle Children's, who's amazing, basically developed a semi-quantitative. It's really hard to develop a full quantitative tool to assess the degree of neuropathology for these kids, but it was more of a semi-quantitative scaling for their neuropathological abnormalities. And the summary of this is, and we're still kind of going through each of these regions in detail, is that there is partial correlation. It's not perfect, meaning that there are regions with a 0% level of mosaicism that still have balloon cells or dysplastic neurons, whereas others might actually correlate more closely with the level. So it's an imperfect correlation, but there is some correlation between the mutation and the neuropathology. And I'll skip this actually in the spirit of time because I wanted to really wrap up about the therapeutic aspect of things. This is the third case, and this is from an out-of-state patient who had diffuse brain overgrowth, as you can appreciate. This poor child had diffuse cortical dysplasia, as you can appreciate, and so he was not a surgical candidate, unfortunately. Passed away at 8 weeks of life. So really severe clinical picture. And this boy essentially haunted me for about a decade because all we got from the team from out of state at the time were paraffin-embedded blocks, basically, from his autopsy. And so the quality of the DNA was extremely poor. And we had tried several things, failed constantly at finding anything and being able to even get some usable DNA. And so, and this is actually also images of his brain showing that he had, such a massively enlarged brain size. It was absolutely fascinating. And he had really diffuse dysplasia. We could never, we weren't able to actually get proper, a full deck of slides from this kid, but he really had diffuse dysplasia as well, of course. So when we started this ddPCR experiment, we decided to throw his samples in there, and lo and behold, we found the same PIK3CA mutation that I showed you earlier. And this is what's interesting. We had a few paraffin-embedded blocks, as you can see here, from these different regions. You'll notice the measly number of mutant droplets we have here, but a solid number of wild-type droplets. So essentially what this showed us is that he may have this PIK3CA mutation at a level of less than 1% of his cells. And this was quite kind of deviating from what we've learned about mosaicism for a long time, which is that we assume that the more severe the malformation is, the more diffuse, the more widely spread the mutation is, and vice versa. That the more focal the malformation is, the more likely it is that the mosaicism is restricted to that region. He kind of showed us otherwise. He showed us that he had bilateral involvement with a very low level mosaic mutation. And granted, this is fraught with technical issues, but we did our best, essentially. We repeated, each of these were technical triplicates, and we ran them 3 times. We've exhausted pretty much what we can do. And we do believe that it's likely real, but we are essentially at a dead end to confirm it even more. But how can such a diffuse brain malformation be caused by a mutation that's present in only 1%? And that really raises a very important question in the field that a lot of groups are asking too, which is that could these mutations act in what's called a non-cell autonomous manner, meaning that perhaps it's not the mutant cells that actually mediate the phenotype. We've seen glimpses of this from tuberous sclerosis with cortical tumors, where there are studies that show that the peritubular tissue actually might be more epileptic than the actual cortical tumors. And there's a lot of literature. I know it's really simplifying a lot of work. But the same question applies here, is that could this be caused by non-cell autonomous effects? Could the mutant cells impact the signaling of the non-mutant cells and mediate a broader phenotype than just by virtue of having the mutation, if that makes sense? So this is, I think, a key question that we're trying to address.
Before you go on, I just have a quick question. Sorry to interrupt you.
Yeah, of
Have you ever looked at normals and screened for these mutations?
A grant reviewer, have you reviewed one of my grants? A grant reviewer asked the exact same question, which is a very valid question. So, no. The short answer is no. Our group hasn't, but Chris Walsh's team has actually been looking at the mosaic landscape of normal brains for quite some time. So he published a paper about a few years ago where they did basically really deep genome sequencing on 3 entire brains. One of them was a trisomy 18 kid, which is kind of an interesting comparison, but control, quote-unquote control brain tissue samples. And they published that there are mosaic changes in the brain. None of them were in the mTOR pathway, thankfully. So none of them were the same mutations that we're talking about. But that's a really important question, and this is why we've been actually trying to collect. So we've collected from the NIH Neurobiobank, from the University of Maryland Bank, any normal brains that are age-matched, region-matched as much as possible. It's not a lot, as you can appreciate. It's really challenging to get. Humans tend to like to hold on to their brains generally, but it's a very valid question, right? Is that we don't know the developmental timing of these mutations. Honestly, we're assaying them at a specific snapshot in time, which is at the time when the phenotypes represent themselves and when these kids present themselves to surgery. So we really don't know when they originate in development, and does the level of mosaicism actually change developmentally? Can some of these cells die off, and therefore we get an arbitrary kind of level depending on which cells have the most survival abilities and stuff like that? These are all really interesting, I think, questions. And that's the level of droplets over here. So the last part, which is, I hope we're no more than 5 minutes, is that it places us at kind of in a very interesting area, is that we've got a wealth of genetic information that I hope I kind of highlighted briefly through this whirlwind tour. And then we have a lot of inhibitors that are developed for cancer. You've got single inhibitors that are shown over here, with rapamycin being a great example. Then you've got dual inhibitors. And then we know that there's this whole spectrum. Honestly, our general view is that mutations of this whole pathway cause a spectrum. And if you want to pick and choose which phenotypes to focus on, that's fine, but there is a very uniform and global picture here. And so we've got an interesting challenge here, is that there's a wealth of genetic information and there's potentially a wealth of drugs to tackle. How do we bridge the gap? Obviously, designing clinical trials for each one of these is not practical. It's a lot of money. Designing animal model trials for these is also a lot of money and a lot of work. And most of the drugs, as we all know, most of the drugs passing through clinical trials fail. They're ultimately not successful. So how do we tackle this? And I think this is one of those really key questions. And we know, and I wanted to show you here, what is some of the momentum going? Of course, tuberous sclerosis, and this is a table that's showing the list of clinical trials, the long list of clinical trials in tuberous sclerosis. It really paved the way, I think, for a lot of the work that we should be doing, which is to design as Jason and I talked about, which is to design properly structured clinical trials for different endpoints, decide what those are. And this is just a list of what's happened with sirolimus and tuberous sclerosis, including neurocognitive phases of the EXIST study for SEGAs, and then for epilepsy and cognitive issues. I think this is kind of a paradigm that we can follow. And so what's been happening with PIK3CA, with the mTOR pathway? Interesting things and interesting developments. For one thing, in a subset of these kids, we know that the ketogenic diet might be helpful. It's not a great fraction, I know, but and what's been interesting about that is that the ketogenic diet has been shown through some studies to have an mTOR inhibitory effect. So could that be a cause? I don't know. And it could be applicable to across a lot of epilepsy types as well. But there is definitely some evidence to show that it actually has an mTOR inhibitory effect. So this is just something that we've kind of kept in the back of our minds as we've moved along. But some of the work that's actually been happening at SCRI, and this is work led by Kathy Millen's lab, who has a mouse model of PIK3CA, and you can see the mouse really has a whoppingly large brain. You can see the noggin here. And the brain phenotype really is recapitulated. There's a large brain size here, ventriculomegaly. You can appreciate that here with cortical dysplasia. Through a collaboration with Novartis, the Millen team got a drug known as BKM120, which was actually in the process of being FDA approved for breast cancer at the time. And so the Millen team treated both the mice that had the cortical dysplasia and did not have the cortical dysplasia with this PIK3CA mutation. And what they were able to see is that the seizure frequency actually was, and this is work done in collaboration with Frank here as well, is that the seizure frequency decreased in the mouse regardless of the brain structure, meaning whether or not their cortex was dysplastic, the seizure frequency responded favorably to BKM120. They were PTZ-induced seizures, of course, so they weren't spontaneous, but still there was a good response. Sadly, and this is kind of definitely a lesson for me, generally speaking, is that this drug then failed to become FDA approved for breast cancer, and it was withdrawn because, and it was found that in kids it can actually cause suicidal ideation. So these are all some of the logistical issues we have to kind of come to face with when it comes to all these drugs, is that there are going to be drugs that completely crash and burn and will never reach fruition. So as exciting as this is, this drug in particular is not going to be a candidate. But it did show that seizures can favorably respond regardless of the cortical dysplasia. And that's an important point because a lot of groups also ask us well, why do you want to think about treating epilepsy? If the brain is malformed, isn't it too late by the time a child is born? This argues that potentially not. Maybe the mechanism of epilepsy is not just the structure of the brain. Maybe it's a channelopathy. Maybe it's pathway dysregulation and things like that. This is the data about the ketogenic diet. Sorry, I kind of skipped over. But this is some of the data showing that how it can actually have a PI3K inhibitory effect. So phospho-S6 staining can actually is used as a marker for that. And a lot of work has been ongoing for that. This is the last part, and pretty much almost the last slide. This is where I think where the excitement really kind of flares up as well. So this is a recent paper that was published through a French group in collaboration with Novartis. This is a drug known as VYL719. And essentially what they did is that they used it to treat, under compassionate care use, unfortunately, to treat 70 kids with body overgrowth phenotypes with PIK3CA mutations. And they showed dramatic benefits where the overgrowth of the body shrunk quite a bit, and there was really significant improvement for these kids. And sadly, what happened is that when they published this paper, they kind of snuck in there that they had a child with hemimegalencephaly who was also treated with this and had some improvement, quote unquote, and then another kid who had neurocognitive improvement without any details. And then the floodgates opened basically for us because then all the families with, whether it's germline mutations or mosaic mutations, we started getting the calls from families saying, I would like this drug for my child. And this wasn't a properly structured clinical trial. This was just compassionate care use, so we really can't compare the data across the board here. But this is where the field is going, I think. So there's a lot of excitement about some of these compounds. And even though the first wave, most people are focused on the body overgrowth phenotypes because these kids are just so visible right from birth, there are very active discussions, and I think the team here knows very well about treating epilepsy using some of these, especially for kids who have failed surgery, if they've really had intractable epilepsy afterwards. So that's it, really. I think we are at an interesting cross-section here in terms of our work. I think we need to have a more streamlined approach between the genetic findings and clinical trials, and this is, I know, something that our teams have been really excited about, but I do think that we have some bottlenecks in this process from going from genes to therapies, but I think these are things we will overcome, I think. And the clinical trial that Jason has led, of course, is a beautiful example of how useful and impactful all of this work can do. The last thing I'll mention is that one of the things we've worked on to address these bottlenecks, because we have so many genetic mutations and so many drugs, is to develop organoids, so mini brains in the lab. So we collect patient cell lines, whether skin fibroblasts or other cell lines. We try to either introduce the mutation or knock it out, and then we actually develop these very cool things that people get excited about sometimes when they see these little rascals in the lab, which are these 3D organoids that mimic, the brain structure. It's fascinating to see. It's a kind of a life-altering moment for me in my boring life. And so essentially, it's really, really fascinating. And what the promise of this is that essentially you can actually develop cell lines from different mutations at the same time. You can try different drug combinations and see. So it's just preliminary data that you can use to inform, animal model trials or human trials. So this is work that's ongoing now over at SCRI, thanks to a lot of help and support from Nino about this as well. And so the take home is that we need to address the diagnostics for these kids better. We are looking at a lot of things at the same time to improve that end. There's a lot of work going on with cell-free DNA from CSF, from kids, to see if we can detect these mutations in peripheral tissues. Everybody's moving towards minimally invasive, obviously, approaches. So we need to kind of keep up with that. We need the molecular diagnostic approaches to be useful for those avenues. Single cell approaches, particularly with minimally invasive techniques now being employed. Early diagnosis, I do believe, does facilitate really important aspects of care for these kids and recurrence risk counseling. And then preclinical and clinical trials using mTOR inhibitors are a really promising thing, I hope, especially for intractable epilepsy. And then there are cognitive issues. That's a kind of a higher hanging fruit, is how to address better the intellectual disability, autism, and other issues. So with that, I'm going to actually skip this. This is my genetics gang here. This is all about, I'm sorry for this, in the spirit of time, to leave time for discussion. These are our take-homes from the digital Drop of Pcr, that it is a useful method. It has some limitations, but we do believe it is useful as a diagnostic panel, actually. So we're working with UW to set this up as a panel right now. And that's it. This is this is a work, honestly, that is really the product of a multidisciplinary collaboration with a lot of amazing individuals that are shown on this slide. So I'm simply the mouthpiece for a lot of the work going on here, and so really thanks to the entire team here, and then of course sometimes the invisible folks on the research side, and this is our lab team and our funding sources, and thanks a lot for your attention.
Ghayda, that was a brilliant talk. I just, you touched on it in the last slide, but it seems like the developmental genes, and you have a mouse model, why can't CRISPR, the gene editing, be used for an mTOR? I mean, is that the future?
Yeah.
I mean, it seems that would be the obvious choice if you have good diagnostics with a single gene.
Yeah. So absolutely, Rich. So on the functional side, for the preclinical trials and stuff like that, we are using CRISPR right now. So actually, this is a key part of that. And because to address the question also about comparing mutant and non-mutant, which is a big fundamental question, because of mosaicism, we need the normal control cells as well. So CRISPR is really a key tool for that. Yeah, absolutely. So we are using it in the lab to generate the control lines, which is, I think, one of the advantages, because unfortunately we don't often get brain tissues that have 0% level of mosaicism naturally. And so this is a key part of that. Yeah, totally.
Thank you.
Sure.
Yeah, I'm going to ask a question. This is kind of out in left field. I know Jeff had an interest in this for a while, but do you think any of the small number of cells that have the mutation could be due to a maternal chimerism?
Yeah.
And it'd be easy to check in the males because you could just say which of these are female cells. And that's like simple.
Yeah.
And then you could ask, are these the same cells that have the mutation?
Yeah, that's a great question. There's a few groups that are working on this part of things. It's a little bit, we haven't systematically done that. And that's kind of like we think of this as like the second wave. Like the first phase we've been focused on is to find as many mutations as possible, as low as possible. So we're trying to optimize the mosaicism part, which is why we focused, we use ddPCR. The second is really to address these higher kind of hanging fruit in a way. But I think it's actually critical. Yeah, absolutely. Yeah. But unfortunately, we haven't done anything just yet, at least our group, for this. But yeah, a few groups are looking at that as well.
Well, I was just going to ask, sort of along the lines of like Dr. Allenbogen is asking, is there any way to diagnose these prenatally so that you could target those pathways before the brain malforms?
I know, right? I mean, wouldn't that be nice? I mean, the problem
Like, do you
is,
see any of this on fetal MRI? Can you see any of the malformations? Or do you know what time what stage in development the malformations occur?
I mean, obviously for like the more severe, the diffuse brain overgrowth, megalencephaly, hemimegalencephaly, they can be identified, right? Vocal cortical dysplasia and the more subtle ones oftentimes are not really detected. I mean, and that's the challenging part, is that for the FCD cohort, we're asking the same questions that folks in the TSC world have asked for a long time, is that how do you diagnose? So for TSC, a lot of the epilepsy clinical trial discussions have been that they want to target the epilepsy even before they have seizures, right? And the nice thing about, not that it's nice, I shouldn't say that, but for TSC at least you have visible features, body-wise, skin, dermatological findings, things like that will actually allow the children to be diagnosed a little bit early. A lot of the kids on the spectrum are only diagnosed when they have their first seizure. So it's oftentimes even when we talk about even a later time point than what you're talking about, which is even at birth, can we diagnose these kids? Oftentimes, from my understanding and my experience so far, is that the answer is not no, unless they present with really obvious like skin findings or something like that. But
how often do
prenatally,
you find mutation in blood?
how?
How often do you find mutation in blood?
So depends on that spectrum, right? So with FCD, virtually never, frankly. With a diffuse brain overgrowth, in about 30% of the time.
Because
If
if you drew blood from the mother, 1 in 10,000 cells is fetal, and then you just fact sort with fetal hemoglobin,
yeah.
and you would have the cells.
Yeah, and there are now, as you know, like NIPT and cell-free DNA testing on, for fetuses, prenatally and stuff like that. The methods are really promising, actually. I mean, and Tina Lockwood and Colin Pritchard over at the U have been doing amazing work with this for brain tumors and other things. I think the methods are coming along, but yeah, absolutely. But it's work. Jason, did you want to say something?
Sorry, I was just going to say that the other compound here that I think Ray was getting at is that in TS, 80% of children will have medically intractable epilepsy, and then about 30 to 40% will have autism. These are known defined phenotypes that will occur across the spectrum of TS. In the enteropathies, it's probably much lower. We don't know. So just because we see the radiologic phenotype of cortical dysplasia doesn't necessarily mean these children are all destined to seize.
Yeah.
And so that's where I think, like to Laura's point, where it's really tricky is that it's actually a lot easier to preemptively treat the TS population
Yeah.
because we know the natural history, but we don't even really know the natural history of how many kids are walking around with focal cortical dysplasia that are completely asymptomatic. How many times have you guys seen an MRI on a completely asymptomatic
Or you
patient?
see people who present with their seizures as young adults or even old adults.
True, that's true
From
too.
cortical
That's
dysplasia.
true too. How many have late onset?
Yeah.
How many have zero? Have no onset at all? Like we don't know. It's a good question.
And this key point, which is so fundamental, I've been saying it to families, it's the last thing they want to hear, unfortunately, is that we don't understand the natural history, unfortunately. It is the truth, and it makes me extremely unpopular when I say to our families that have been calling and saying, I mean, imagine the challenge is that right now we get calls, I mean, and this is not just our team, I mean, I think it's happening everywhere, is that families are requesting these drugs now under compassionate care use for this indication or that, when we still don't even understand the natural history. And so I think it's an interesting challenge.
Manny?
That was terrific.
Thank you.
You talked about germline disorders and 50% allele frequency, and then the spectra of mosaicism, and then at the other end is going to be completely somatic, sporadic events,
Right.
like cancer. You
Right.
alluded to specific genes, specific hotspots being involved with cancer and your syndromes, and these children don't develop tumors or cancer.
Right.
Very rare.
Yes,
So
that's true.
how do you resolve that?
Yeah.
Is it a cell-specific
Fascinating, yeah.
phenomena? Is
Yeah.
it pathogenic, allele frequency?
Yeah.
What's your
So for the longest time, by the way, like people were screening the kids with the germline mutations for Holmes tumor, and there was a whole franticness in the genetics world about could these kids be at risk for cancer that we're missing? And that really terrified people, I think, for a long time. There was no data to support whatsoever of the limited natural history data. Because again, what happens is that there's a peak of publication. I mean, the bias is that you publish to describe these new syndromes, but then hardly anybody is incentivized to publish long-term natural history data. So there was no data to support that there was an increased cancer risk. So the way we've kind of consolidated, or our view is that, first of all, the differences between at least these developmental phenotypes and cancer is that in cancer, these mutations are hardly the only mutations, right, in those tissues. So PIK3CA mutation, which is among the more common ones in somatic tissues in cancer, oftentimes it is a driver mutation, and then you get a lot of other passenger mutations in cancer tissues. Whereas with these phenotypes, and even though, again, we hardly do like whole genome sequencing to look across the genome for second hits and whatnot, and that's a really key point. Unlike the cancer groups that have really paved the way to doing better genomics than we do, well, generally we still don't have an idea that they have a second hit. Usually from what we've seen, it's this one hit that drives the phenotype. The lesions look very much, I mean, certainly the body lesions, if you look at them, they look a lot like cancer. I mean, they're just, I mean, and they're invading and they're crossing fascia and there's an invasive thing about them, but we really don't know. And I think this is why what we're trying to do is really, I can follow the footsteps of cancer. We want to do high-scale genomics. I just submitted a proposal to say, like, even in the mutation-known kids, we should genome sequence them to see if there are second hits somewhere, you know. And we should also do some of the same pathway abnormality, like proteomic approaches and RNA sequencing approaches that the cancer world has done to understand the pathway as a whole in these phenotypes, because we haven't done that. And so, but I think that's a really important question, actually. Yeah. But I think the key thing is, again, the differences so far. It appears to be these are isolated events, which for pharmaceutical companies, they love that because then you're giving them a pure model, essentially, versus cancer, which is extremely molecularly complex and such.
Do you have any cell-specific data on those regions of the brain? Like
That's-
where the...
Yeah, that's my dream, to be honest with you. No, we've been trying to fact sort, you know, cells and then do, you know, so single cell methods basically to get specific population. We're still in too early of a stage to even, you know, it's fraught with some technical challenges. But the nice thing is that single cell RNA sequencing now has been really on the horizon with the BBI, actually. They've been pushing for it quite a bit and making huge developments. We can potentially use that to isolate the populations, and then we'll need to do the genotyping on those cells. Yeah, it's a key point. We don't know if neurons or glia are actually necessarily the ones that are mutants most of the time.
Jeff, as chair of surgery and executive vice president, you get the last word.
I do. So the male's brain actually is maximum at age 12 in size.
Are you asking?
So, no, it's a statement. That's a statement.
Well, am I supposed to
No,
comment?
it's a statement. I have a question. Well, first, everyone, think about the implications of that. But, where do these? How do they play in normal growth?
Yeah. So again, this is all the tip of the iceberg, Jeff, right? Like all we see is the tip of the iceberg usually. The degree of brain overgrowth with these phenotypes in the first 2 years of life is so dramatic. I mean, actually, a lot of the kids actually, even in the old literature, they would get even preemptive neurosurgical shunting, even if they had like mild ventriculomegaly at the time, because the degree of brain growth was so dramatic. So far from what I've seen from the hundreds of kids that I've seen with the megalencephaly, the diffuse brain overgrowth, and this is all anecdotal data, I think, but the maximum period of brain overgrowth is the first 2 years. They shoot up, they cross all the percentile points essentially, and shoot up and reach a point above the growth chart. Afterwards, we've noticed that generally they tend to then follow the general normal growth trajectory for that age group and then that gender. So
But do we know much about the role in normal brain growth?
for these genes? Yes and no. So so the problem is actually, this was a fascinating thing which pushed us in the organoid direction, is that there was a paper recently that showed that the mTOR pathway actually is specifically activated in primates and beyond. So meaning that it actually, and it's one of the major drivers of forebrain development and the dramatic expansion of the cortical volume that occurs in humans and primates. And the problem with that is that unfortunately mouse models in that sense might not be the best, because then it actually is not activated in the same fashion in mouse models, and obviously mouse brain is not human brain. And so it is believed to be one of the major drivers of neocortical expansion in humans, and that was work comparing human and mice and monkey brains, macaque, and they developed organoids from all three of them, and they showed that using RNA-seq that the mTOR pathway is hyperactivated in the macaque and the human at a dramatic rate. So it is believed to be a driver of especially forebrain development in the normal context, but that's the extent that I'm aware of in terms of its role. So it is a big driver though of brain size. Brain development, in terms of like why does it cause the malformations? Because it causes FCD, it can cause PMG. We really don't understand why it can cause PMG. Is it more of a vascular thing, versus an actual mechanism for PMG, which is very difficult to parse out. So its actual role in neuronal migration and maturation, we still don't understand. Yeah.
Thank you, Ghayda. That was
Thank you.
fabulous.